Hi, my name is Manuel. I'm the second-year student of Traditional Chinese Medicine at UTS. I'm here to present my topic about glutamate and its regulation. The presentation is divided into 4 parts. Introduction, glutamate receptor, effects of glutamate and its regulations. Conclusion. The total time taken is about 6 minutes. Chinese restaurant syndrome is the unpleasant adverse effects such as nausea, dizziness, and headache after a meal with excess amount of MSG, monosodium glutamate. And glutamate is the active source of glutamic acid, one of the lateral occurring amino acids. Glutamate is also an important excitatory amino acid. Excitatory amino acids are amino acids which act as neurotransmitters. They can cause hyperexcitation and even death of the cells. Glutamate is the most abundant EAA and it exists in greater than 90% synapses. Glutamate receptors are widely spread in our body. Apart from nervous system, it exists in other internal organs and tissues such as GI tract. There are 2 types of glutamate receptor, ionotropic receptor and metabotropic receptor. The former are fasting ion channel, the latter G-protein coupled receptors. This is the diagram showing different types of glutamate receptor, and the ionotropic receptor is classified according to the ligand it binds. So, the ionotropic receptors are classified as AMPA, kinase, and NMDA. And recently, we found a 4th type of receptor called delta receptor in cerebellar Purkinje cells. This diagram shows the 3 main different types of ion channel. AMPA and kinase as ion channel of sodium, while NMDA is the ion channel for sodium and calcium ion. This is the synapse in which the postsynaptic cell contains 2 types of receptors, AMPA and NMDA. When the concentration of glutamate is low, only the AMPA receptor open with an inflow of sodium ion, causing depolarization of the cell. When the glutamate concentration is high, the sodium ion is higher in this cell and repel the magnesium ion which originally blocked the channel. As a result, more sodium ion together with calcium ion will rush in. The calcium ion will act as a secondary messenger and trigger long-term cellular change. One of the change is cell death. If the NMDA receptor overstimulated, the increase of ion itself will cause cell death. It also activates the capain and increase the nitrogen oxide and cause the death of the neurons. So it's important to regulate the glutamate in the brain. Ingested NFG can hardly affect the brain simply because glutamate in brain is higher in concentration than glutamate in blood. Thus, efflux is greater than influx. The second is glutamate is charged. It cannot pass the lipo-blood-brain barrier. Third is glutamate is effectively removed from the synaptic clefts by various EAA transporters. In this diagram, the color circle represents different EAA transporters. Recently, we found a new one, EAAT4. All of these are used to regulate the glutamate concentration. There are reactions to regulate the glutamate in brain. Glutamate can be changed into glutamine. Also, glutamate can be changed into alpha-ketoglutarate glutarate. These two are reversible reactions. And the last one is that glutamate can be changed into GABA. It seems that this is not reversible. Actually, GABA can be converted back to glutamate via the TCA cycle. This is a simplified diagram of reactions in brain. It's worthwhile to mention the antagonistic action between glutamate and GABA. The former is excitatory and the latter is inhibitory. Simply speaking, glutamate causes hyperactivity of the cell, whereas GABA hypoactivity. For instance, glutamate can cause anxiety, where GABA causes calming. Conclusion: glutamate relates closely to the brain damage, so its regulation is important, especially in brain diseases. Glutamate and its metabolic product GABA act antagonistically. Their balance is important to the normal brain functions. That is the end of my presentation.